So what I really wanted to discover was I wanted to try to make this connection between disease phenotype, like what a patient presents with, and then also the genetic basis of it. So that is basically getting rid of nuclear lamins, changing nuclear lamins genetically, and seeing what happens.
Now, I'm curious to hear about the work that you're doing in terms of, like, what are you researching in sort of in basic terms?
Very interestingly, like very recently, I think it was yesterday actually, there was a New York Times article that came out about a genetically caused disease called Hutchinson-Gilford progeria syndrome. And this is a premature aging syndrome. And what happens is that these patients, they all have mutations in one gene that codes for a nuclear lamin, which is a building block of the structure of the cell nucleus.
That's what a nuclear lamina is. It's a building block of a cell nucleus.
Yes, so
Okay.
the structure like think of the if you think of the nucleus as just like this um filamentous thing. There's a structural scaffold that keeps it in like round and holding the DNA within it. And that is called a nuclear lamina. A lamin, Lamin, is each of the building blocks of the lamina.
So it's like a brick in a brick wall that makes
Exactly.
the building.
Yeah,
Okay.
exactly. And so people who have mutations in the genes that code for nuclear lamins, they have diseases. And one of these diseases is a premature aging syndrome, where a lot of these patients, they show signs of thin skin, frail bones, cardiovascular disease. Most patients they have symptoms of like very aged, like 95-year-old patients when they're only 13 years old. And so it's a devastating disease. There's no cure for it. And a big question is why would a mutation in a gene that codes for like a building block of the cell nucleus lead to a disease with so many phenotypes? Like you have cardiovascular problems, you have muscular problems. It's awful, really. And so what I really wanted to discover was I wanted to try to make this connection between disease phenotype, like what a patient presents with, and then also the genetic basis of it. So that is basically getting rid of nuclear lamins, changing nuclear lamins genetically, and seeing what happens. And so our hypothesis going into this was that we hypothesized that weaknesses in the nuclear lamina would lead to something called a nuclear membrane rupture. So that is exactly what it sounds like. It's the nuclear membranes basically having spots of weaknesses, and then you have the contents of the nucleus, which is the DNA, spilling out of the nucleus and then getting exposed to all of things in your body's that in the cell that it's normally not exposed to. And then we suspect that this process then incurs a lot of DNA damage, which then leads to the phenotypes, like the premature aging, the the disease phenotypes that we see in real patients with mutations in their genes for nuclear lamins.
It's like the wall of the nucleus is breaking down, the genetic code is spilling out of that, which has tremendously awful implications for the person whose body that is happening. Um,
Right.
and that is connected to this disease that these people have,
Yes. And not
resulting in basically their body like degenerating.
right. And not just that. I was just naming like one example of a disease caused by mutations in nuclear lamins. There is a wide variety. Like I'm actually like one of the things that I ended up focusing on studying a lot was microcephaly, because it was found that patients. Sorry, microcephaly is like abnormally small
It's
head
it's a
brain.
small head, right?
Right, exactly.
Yeah.
And so one of the things that people have found was that mutations or missing one copy of a gene for a lamin led to neurodevelopmental defects. And so I ended up like looking a lot into this and doing studies with neurons, neuronal models that I grew myself, and examining what would happen if I eliminated the nuclear lamins from these neurons.
Okay. Um, so I'm going to back up for a second. So the neurons, right, which we think of neural links, these very, very long, thin cells that connect and send signals from our brain all through our body. You're studying specifically neuron cells. So where there's a nucleus inside of a neuron, and it's in those type of cells where problems with the nucleus are happening that specifically might relate to microcephaly rather than like other types of cells that are not neuron cells.
Exactly.
Okay. What was the problem again, specifically with the neuron cell that was resulting in microcephaly?
So that is a great question. That's like actually a big part of what my research was on. So when your brain forms in embryonic development, so when everyone is still like a fetus, your neurons form by migrating. Like your brain starts as just one single layer of neurons, and then over the course of development, these neurons divide and then they migrate to form layers of a brain, basically. And what we hypothesized was that so these neurons are migrating. Migrating cells are subject to a lot of mechanical forces because there's movement of the cells involved. If a neuron is migrating and it has a weak nucleus, like it has a mutation in a nuclear lamin that makes its nuclear structure quite defective, this migration process probably will then lead to a lot of DNA damage incurred inside the nucleus because the migrating cell then pushes against the nucleus. The nucleus is already structurally weak. This causes there to be ruptures of the nucleus, exposing the DNA to your cellular contents, things that they're just not supposed to be exposed to. And then this might in turn cause DNA damage, which then makes the cell damaged and leads to defective migration of neurons and perhaps then defective brain formation.